Welcome everyone to another episode of the Beyond Terrain podcast. I am your host, Liev Dalton. If you're new around here, consider subscribing or following the show. And if you like what we do, consider liking, commenting your thoughts on the episode, leaving us a rating or a review. Sharing is always the best way to support us. I'm thrilled to finally announce that the Beyond Terrain community is live. It has been a long time coming, and this is more than just a platform. This is a representative of a lifestyle shift. Beyond Terrain is a movement. It's part of a bigger paradigm shift, the terrain paradigm. And this is a place to unlearn, connect with others, learn from others, and reconnect in a meaningful way. Rooted in nature, guided by logic, and driven by truth. It is free to join the community, so there's no reason to not sign up. You'll gain access to introductory classrooms on the causes of disease, as well as master classes and the fundamentals of the terrain paradigm. If you want to go deeper, becoming a full member gives you access to many more things, including deep dives. First, we have dove into parasites and covered absolutely everything about them. Methods used in science, their ecologies, human experiments, etc. Future deep dives will include cell biology, genetics, mold, bacteria, etc. We have absolutely no limit. We also discuss things like movement, lifestyle, reflections. We do live Q&As and discussion calls. Of course, as a member, you'll gain access to future discussions, live calls, master classes, workshops, etc. You will also have the opportunity to win terrain-approved prizes. This could be organic, natural clothing. It could be books. Our founding member offer only contains 100 spots, and these will go very quick. You'll be able to lock in that low price, gain access to a complimentary onboarding call, and what you'll really gain access to is shaping the community. This is a growing, living, breathing community in which you'll be able to support me and my work while getting lots of value in return. So go check it out. But without further ado, let's get into today's episode. Dr. Tomsic, thank you for coming on the podcast once again. Really looking forward to our conversation today.
Hi, Liev. Thank you for contacting me again. I think we promised, right, some sort of continuation to our PCR discussion. So yeah, thank you for having me.
Of course. Yeah, so if you guys haven't checked that one out already, this is our second podcast together. Make sure to go listen to that first one. It was all about PCR, dismantling that. We'll probably touch on PCR a little bit today. It's really a central topic when it comes to our main topic of discussion today, which is the genome, DNA, genomics. What are we even going to be talking about today? There's lots of questions going on. We're going to try to address some of those questions. This is kind of a difficult conversation because I'm sure Dr. Tomsic will agree that it's a little bit hard to discern what exactly is going on if, DNA doesn't exist as we know it, so trying to provide some sort of positive statement, of what's going on is the more difficult thing to do, right? So we'll try to stay within what can we objectively observe and confirm, and then question the rest. The reality is, I know those folks like Dr. Cowan and Alex Zeck, they always discuss these things that you don't necessarily need to provide a positive claim when you falsify a claim. That's certainly the case, but it's nice to maybe speculate a little bit and try to discuss what exactly is going on. Jerneja, is there anything that you want to open with here, leave the listener with before we kind of dive too deep in here into this topic?
Oh boy. I mean, of course, I've been accused of somebody that has no clue what she's talking about, right? Because it's completely the opposite compared to what I was told in school. So yeah, it's hard for people that are just jumping in and hearing it for the first time. It's hard for them to completely grasp it, or at least to say, okay, maybe she knows what she's talking about, but it's completely the opposite of what I was told in high school or whatever, if somebody did some college classes of molecular biology and stuff. But what I realized 5 years ago, having an open mind is the most important thing going forward. And it's only served me well, because if I closed it back in the day, I wouldn't be where I'm at. Meaning I'm still working in the same job, working with DNA that doesn't exist. So, still chasing down in -80 freezers my DNA samples and really wondering why I'm still there. But I know that I have to be there because there are so many changes going on right now. So just a brief thing that I want to throw out for people that are not in academia, that are not around professors, that are not in these high important places, whatever. The amount of going on is a lot because research funding is being cut.
Mm.
And I'm happy about it because once I understand what research is, how we are going down completely the wrong path, like completely the opposite, completely upside down. So I'm like, okay, once I understood that things need to change, I'm like, how are they going to change? Oh, they're going to change the hard way. All these different professors will stop getting funded. First, it was based on, oh, if your research is on diversity, equity, and inclusion with that whole thing, then you will stop getting funding. And now it's going further and further into things where even my supervisor, she has no clue what's going on. She just believes that everybody that's in charge of cutting the funding is completely incompetent. So this is the point of view of somebody that has truly no clue what's going on in the world, that truly has no clue that everything is upside down. For her, everybody that's doing what they're doing are just incompetent. So, but I think I need to be the witness of the whole madness. And in the meantime, I'm talking to a few people, a few of my coworkers, and explaining things to them why things are the way they are. So they are not completely lost because they are less brainwashed. They are more fresh. They are younger. They just finished maybe grad school or something. So you can still influence them in a good way, in a positive way. Like explain to them, look, try to find something that has, that has future, let's say. But my supervisor, who's way over 60, how am I going to change her mind? That's just not going to work. But at least maybe I can drop some seeds, here and there. So that's just one thing that I wanted to throw out there because, I mean, people that are not in sciences, they don't understand why some things are going relatively slow. Why RFK Jr. that took away those advisors, the CDC advisors, put on some of the ones that are questionable. I definitely don't like how Dr. Malone is doing stuff. He still supports mRNA, whatever vaccines, but he's kind of saying, I believe he's saying they are not for COVID, but then for the rest still could be okay. So I understand why it has to be this way. Because if you completely change it to people like me and like you and like Dr. Cowan, no, there would be a lot of heart attacks in academia. A lot. I mean, like people would be falling over because that would be just too much. So just starting with this. So
Perfect. Perfect. Yeah, great way to get it going. Yeah, I think that, dogma really has no place in true science and true academia. And so it, like a lot of these things were really difficult, like starting with the germ theory and now moving into DNA. I'm encountering a lot of resistance when I talk about these topics. Even people who are against, virology and germ theory, now it's like, well, no, but DNA does exist. We know what DNA is, right? So it's so interesting, how we still have our walls up. But I think it comes back to like, and we're gonna evaluate some of this stuff today, but like what's actually going on? Like what are we actually doing and looking at? Because genomics is so deep in the test tube. It's past the test tube, right? A lot of genomics takes place on the computer, right? So we're so far removed from reality. When you look at what's going on in the laboratories, you're working with gel electrophoresis and PCR and these machines and electron microscopy and X-ray crystallography and chemical reactions. Like what does this have to do with life? What does this have to do with our biological systems? And what's the validity of these laboratory techniques to what goes on inside of our bodies, right? There's obviously, the resistance I get it. I get it's hard maybe to rethink some of these things. But at the same time, like you said, that curiosity, that open-mindedness, I think is so important so that we can continue to adapt moving forward. Genomics is plagued with circular reasoning. There's no foundation whatsoever. Lacks the use of the scientific method, as we'll see today. And I think really the resistance comes from the theory ladenness, right? The idea that the genomic and DNA language is in our everyday use, right? It's not just in academia anymore. It's like now when you're out on the street, it's, oh, well, that's in my DNA. It's common language, right? So, and I hate this term, but for the lay person, to understand the concepts and to attribute it to DNA, was very easy because it gave us that piece of understanding. Like, yes, okay, I have an answer. I know that because I look like my parents, it's because of DNA. But the objective observation here is that you look like your parents, it's not necessarily because of this DNA in question today, right? So that's kind of the whole point. Like no one's denying these objective things that we can observe, right? It's a matter of questioning sort of our theory, our explanation, our stories that are told about what we're actually seeing. So I guess it's probably a good time to get into the discussion. Maybe I can give you the floor to discuss DNA. Why are we even questioning DNA as a double helix model? And, where does this even come from? So I kind of want to just give you the floor wherever you want to take it and start. I'll give it to you.
I mean, why are we questioning it? I wouldn't be questioning it. Back in the day, I was just going along with everything and isolating DNA, isolating RNA, and I was questioning nothing. That was, I mean, after a while, you just go with the flow, right? Who are you to be questioning stuff that was done in early 50s, late 40s, whatever, back in the day? You were not even, I mean, not even your parents were born, probably back in the day, right? So my whole thing, so I started questioning PCR. My really starting to question science was when I started questioning PCR and how that was used for diagnostic purposes, diagnosis. I just recently came up or found out research papers that are talking about how PCR is problematic even to diagnose pathogenic bacteria and all of that. So it's not just viruses. We know viruses don't even exist. So because there is no proof they've ever been isolated, right? But let me just share, because that's usually how I do things with my PowerPoint slides and stuff. But let me just share why I actually started questioning DNA. So I was sent this blog, whatever. It's not a peer review paper. Again, we are not going to discuss problems with peer review. People should stop requesting me to supply them with a peer review paper because that's all a bunch of nonsense also. And it's been stated by journal editors for 20, 30 years. It's been discussed, the peer review process, how that's a problem. So Tamra actually wrote this, is it a paper? Is it a post? Whatever. Yeah, so DNA discovery, extraction, and structure, a critical review. So I'm not gonna read it or anything, and I'm sure Liev can post, you'll have the link below.
We'll put all the links down below, yeah.
So this was sent to me by a couple of good friends that I trusted with a lot of other stuff. So, over 2020, I pretty much came across both of them in 2020, and I realized they are trustworthy. So when they sent me this in 2021, oh yeah, so this was December 21. So we've had a couple of years to really become close friends. So when they sent me this, and I remember it was a few days after it was published, before Christmas, and I didn't even open it because I saw the title and I'm like, oh boy, now I'm gonna be questioning everything that I've ever done in the lab. So how was that gonna go? But then few months later, they touched base again because they are not scientists, and they wanted me to explain a few things to them as a scientist that kind of understands few things maybe a little bit better. They were like, Jerneja, we really want to learn something, and we need to talk. So I'm like, okay, let's do this. Let's jump in. So Tamra brings up DNA extraction. So the first DNA extraction was done. I'm just gonna go briefly through a couple of things. You guys can read the whole thing if you want, but when you don't have the knowledge, I mean, whatever that knowledge is, but when you at least don't have that knowledge that they gave us in molecular biology class or whatever, it's kind of also hard to understand, although she writes it very much for the layman. So it's not that hard, but still I want to make some comments. So this Swiss physician, he extracted it. So he isolated, I put these question marks again. So what does it mean, isolate? How did he do it? Everything is a little bit questionable. And he was isolating DNA from leukocytes, white blood cells, because this guy, a biochemist and lab owner, Hoppe Saylor, he suggested he goes and grabs bandages from wounds. In bandages, there is a lot of pus, there is a lot of blood white cells. So he said it's going to be easy to isolate DNA from there. He determined that he obtained a novel substance that might be nuclein through the following observation. So what he was actually doing, he was mixing a lot of stuff with those cells. I mean, just chemicals. He got precipitate, and then alkali was dissolving it. So he just decided that because it's alkali is dissolving it, then it's an acid. The isolation procedure produced almost zero amount of sulfur, a byproduct linked to protein, but a high amount of phosphoric acid. So again, it was assumed it had those phosphorus groups. And basically, he concluded that he has discovered a novel substance based on the reaction of the obtained solution to the chemicals used and produced employed, and not based on actual isolation and observation under the microscope of the content of the nucleus. Because under the microscope, you cannot see anything. So just based on, it was not smelling of sulfur, it has phosphor, so it contains phosphor. So everything based on what do you call it, like secondary evidence. He didn't really see nothing. He didn't see nucleic acids. So these are nucleic acids that are components of DNA. They were discovered, or they were kind of determined at the end of 1800s by this other guy, a chemist, Albrecht Kossel. Adenine, guanine, cytosine, thymine, and uracil is in place of thymine in RNA. And again, how did he discover this? She's kind of putting it out there. It's really hard to go and find original papers. And then even when you find them, it's not really clear how all this stuff was discovered. But then again, he was awarded Nobel Prize in physiology or medicine in 1910. So what we are noticing with all of this stuff, that everything that kind of has to be confirmed, like I call it in the material world, because this is what it is, right? This is the material world. DNA is something that theoretically you can touch. They just present it to you as, yeah, you would be able to touch it if you had something that is small enough that can touch it, but you really cannot. I mean, you can touch a tree, right? But not the DNA components. But everything that needs to be, because earlier you mentioned stories. You said something, I didn't write down the exact, the stories the scientists tell. So these stories have to be blessed by somebody, and usually it's the Nobel Prize committee that blesses all the stories, right? And we know it's the viruses in the 50s and all of those stories, how they've been blessed again by the Nobel Prize committee, and so on and so forth. So then she brings up, I'm going to say, so this is all out of that paper. So Tamara did all this research. I'm just
Yeah.
going off of it, right?
Can I just mention something really quick, actually, just about that last slide? Um, what kind of popped into mind, and I don't have an answer for this, but when you go back and you read, and it's quite elusive to try to find the methods used in which he isolated these base pairs. It's very, very difficult to find the actual methods being used. One thing that we should question is how it was determined that one was from DNA and one was from RNA. How did he supposedly isolate the two and determine that uracil and thymine were categorically different in that manner? Right. So it's questions like this that like absolutely make no sense. First of all, when you talk about the initial isolation of DNA, you're talking about burned, acidified, centrifuged, spun, precipitate, and then we just call it nucleon. We just call it DNA, right? We just say, yes, this is the basis of life. And they say, oh, it's phosphorus rich. So that's the objective observation that he knows. It was phosphorus rich instead of sulfur rich. But he added phosphoric acid to isolate it, right? Kind of a hilarious little confounding variable there. And of course, if you don't add phosphoric acid, if you don't add it, you don't get DNA. So how does that process, how is it controlled whatsoever, right? How do you know that the substance, first of all, we don't know anything about this substance. We just have a little precipitate. How do we know that that is the same thing that is in our body, right? This is what we're talking about when we say uncontrolled, right? Unverifiable. How can we verify that? So I'll let you continue with X-ray.
Uh, Perfect. Thank you for pointing all this out because this is important. This is what, this is what I was not thinking about back in the day when I started college. I was not questioning. I didn't go back to the papers. How was this done? I just assumed somehow they figured out uracil and thymine. But again, this is a good question. How? Who? When? So yeah, very important. So the DNA structure, this was 1952. So this is what they call, from here, it's clear that it's a double helix. Oh, Lordy, I don't know. This is what they say, right? So this was published, this is the famous photo 51. Again, she goes deeper into explaining why this is problematic, but just one needs to look at this picture and understand that this is what's being called, based on this, we know it's double helix. Again, where do they see that? Nobody knows. And then on April 25th, 1953, the famous journal Nature published 3 articles about DNA. So the most famous is this, A Structure of DNA, by Watson and Crick. So this is like the most important piece of the DNA Bible that a molecular biologist knows exists, but usually never reads, right? So there were other 2 papers. Wilkins was one of the, I think, collaborators of Watson and Crick at some point. He published this paper, Molecular Structure of whatever that is. I really don't care what's going on there. And Molecular Configuration of Sodium Thymonucleate by Franklin. But if we pull out the paper, probably I read this paper back in the day, like 30 years ago, or I guess close to 40 years ago. I don't know, but I think 30 years ago, when in college. And probably I never noticed this. We wish to suggest. I didn't underline all of this. Oh, actually, I have a little bit bigger picture over here. So 1962, again, DNA structure that was just a model, but was proposed, they suggest. And you guys can stop the video if you want to read this. And the words that are being used, are just completely ridiculous. There is no experiment, no nothing. And again, they get the Nobel Prize, and this was Watson. Crick died, I don't remember, quite some time ago, but Dr. Watson is still going. I met him in Cold Spring Harbor Laboratories on Long Island in 2007. My husband has a picture with him, so he's been around for a very long time, and he's still around. So just saying. A lot of stuff. So they based their model on the Chargaff rule. So the Chargaff rule, somehow he determined that the percentage of A is equal to the percentage of T, and percentage of C equals to the percentage of G, because A and T bind together, make the base pair, what's called, and G and C make the other base pair. So this is how, because this is, the percentages are the same, so this is what it means. I mean, again, it's all deducted out of, because this is how it is, so it has to be this. And then he actually found that the percentage of AT and GC changes based on different organisms. And what was kind of found when sequencing bacteria, that bacteria that live in at higher temperatures have a higher percentage of GC. So again, why is this so? Is it true? Is it not? As Liao said, right now I'm at the point of finding what is not right, what is not true, and why. Finding the other way, like really trying to explain what. This is my project for my next decade, I guess. For right now, I'm kind of dismantling all the lies that I've been told and that I bought into as if there is no tomorrow. So I found, so this is Erwin Chargaff. He died in 2002, so he was of a decent age. And the interesting thing is, again, I'm not going to read this, but the very interesting thing is, beginning in the 50s, Chargaff became increasingly outspoken about the failure in the field of the molecular biology, claiming that molecular biology was running riot and doing things that can never be justified. So he actually even wrote a book published in 1978. I haven't read, it's on my to-read list, Heraclitean Fire of a Molecular Auschwitz, that the technology of genetic engineering poses a greater threat to the world than the advent of nuclear technology. So just saying, an irreversible attack on the biosphere is something so unheard of, so unthinkable to previous generations, that I only wish that mine had not been guilty of it. So it's interesting. A lot of people think that scientists are evil. They're just evil, and they do all this evil stuff. No, we are just a little misled and misguided. We do things that can be used for good or for bad. This is always the thing. Like, whenever something is discovered in a scientific way or not scientific way, whatever, stuff can be used for good or stuff can be used for bad. So when he realized that stuff was used for bad, he was actually sorry that he ever discovered anything or that he ever was part of it, right? And here he brings up, which later on we are going to discuss. I don't know if this is going to be the podcast number 3. I don't know, because there is a lot to talk about, right? But there was the whole thing, because here, 1978. So I have a slide where, right now I don't remember, but in the 70s, what is it, 75? The first in vitro baby was born in the UK in, I believe, 1975. I will see eventually if we find that other slide. So he was already aware what was going on in vitro fertilization, which, yes, it's a good thing. Helping a few couples condemned to childlessness towards getting a child may strike the obstetrical cytologist as such a laudable step. But we can see the beginning of human husbandry, of industrial breeding factories. Who can deny the scientific interest attaching to the production of chimeras, to the study of human embryonic growth in an animal uterus? I mean, this is all stuff that is already going on right now. So he kind of saw it probably already a little bit back in the day and was hoping it wouldn't go that way, but it still went. So that's just an interesting fact about Chargaff. I mean, his rules were used, but then he was even sorry that he came up with those rules at some point.
On the
Yes.
note of the double helix, because obviously all of this is playing into this whole concept of the double helix at this point. I'm going to share my screen for a moment, so you might have to reshare after.
Yes.
Um, some pushback that I got, was that we have now taken a picture of DNA, of the DNA strands, right? Without X-ray technology. Because obviously they blasted, this X-ray crystal or this supposedly DNA salt crystal with X-rays for 62 hours. Well, X-rays are supposed to cause DNA damage, but we'll forget about that for a moment. But now we have this, transmission electron micrograph image of DNA. It's not this blue one here, and it's not this purple one. These are computer generated images. All of these models, they're all computer generated. This is the photo we have now. As you can see, this very staticky image, black and white, showing very subtle helix structure. Again, this is what the evidence now is for the structure of DNA. It should be noted too that this is not the double helix. This is a more condensed form because, through our stories that are told, DNA condenses itself into a chromosome, right? And it packs itself around, these through methylations and through all of these different processes that, again, are all backfill and nonsense that has never been proven. Any of this has not been proven. This is the only evidence we have of a picture, and we know electron microscopy completely alters the sample when you are taking a picture. It's admitted that this is not the double helix structure, because the electrons would break the double helix structure. Again, a claim that is not backed up by any evidence because there was no double helix in the first place, right? So it's these ideas like this that are completely unscientific, that are laden throughout the entire literature. You can't blast DNA with electrons because it will break the DNA strand. How do you know? What experiment has been done? How have you controlled the variables? None of this has been controlled. None of it uses a scientific method. It's hearsay. It's nonsense. It's unfalsifiable for the most part, right? So what we're end up with is this picture, and it gets validated by, the gospel, by the scientists and the communities, and then it becomes the true story, right, through the populum. But I just really wanted to bring this up as, the supplementary evidence of the double helix, right? And I'll link the paper for this as well. It's just, no real use of scientific method, because we're not beginning with an isolation ever either. They're using the so-called isolated DNA that we've been discussing to get this picture in the first place. And then they add it to this, this, I think they call it like a hyper hydrophobic surface, or like a super hydrophobic surface. And it's made out of these little pores and, these like protruding elements as well. And then this right along here is supposedly the strand of DNA that is condensed going across. Like this is the evidence that they are sharing. This is exactly what they're sharing. This image itself is what these papers are being written on. And the whole discussion section is all based on speculation, because what we have is that image of the pores, we have this image, and then we have, I might not be able to find it here, Rosalind Franklin's image that Jerneja pulled up there earlier, looking kind of from the top down, and then they assume the angle, and then all of a sudden you have, double helix. It's just, there's no foundation, right? There's no like, here, look at this. People take these pictures and they say, here, look at this. Here, look at the DNA. Look at the strands of DNA. But it's like, that's not a foundation. It relies on piles and piles and piles of assumptions and uncontrolled scientific methods. Who knows what we're looking at? Nobody knows what this image is. Nobody knows what this electron microscope image is of. We claim that it's DNA. We took a picture of something. We took a picture of a concoction of chemicals, of burn acidified chemicals blasted with electrons. We know that, so that's what we have. That's all that we have. Anyways, I just wanted to add that because that's been something that's brought up over and over. It's like, yes, we have pictures of DNA now. This was in 2012. No advancements have been made since. I don't even know if this has been reproduced. I didn't go that far as to look, but so I can't claim anything there. Anyways, I'll let you continue.
But now, because before I continue right there, I'm going to jump to something, because you were talking about UV light or whatever, X-rays nicking the DNA, so breaking the DNA. There is one interesting paper I want to bring out of a friend of mine. So again, a friend of mine since 2021. Like, I have all these good friends that came with this whole COVID stupid business. So Dr. Fabio Franchi, he is a retired infectologist. He's a doctor in Italy, and he was speaking up against HIV/AIDS lie back in the 90s. I don't know, maybe even in the 80s. Luckily, he was never fired. They only asked him, because he was in the hospital, he was working in the hospital, but they only asked him to not be talking about his quote unquote conspiracy theory nonsense in front of patients. They didn't really say, we're going to fire you, so whatever. So when I came across him on Twitter, I connected with him because I'm from Slovenia. So when I went back, he lives really close to Slovenia over in Italy. So we actually met in person. This paper that he wrote with another professor from South Italy, Roberto Serpieri. Roberto is more of an engineer, so he can actually look into equations and all of that stuff that I'm like, oh, that's a little bit too much for me. So they were asking, is polymerase chain reaction fully reliable? So this was published in a preprint. I don't know when they published it, in 2019. I mean, it's been sitting there for a while when I came across it. A critical review in the light of published evidence from the golden decades of molecular genetics. So what happened, actually, the date to accept it, 29th April 2024. So a little bit more than a year ago, this paper was published. It was a little bit like, some things were a little bit changed. They changed the title. But their conclusion is actually this. And when I read it, I didn't read the whole thing. This paper has an incredible number of pages. I mean, this paper is, stupid long, 1 to 31. So 31 pages. I'm like, you know what, Fabio? I'm just going to read some things that are that I can even understand. And I immediately found something. I clearly immediately found something, and they pointed out. So at some point they see so I noticed pre-1950s papers are talking about DNA as being not very stable, as being broken up when heated at temperatures above 95 degrees for longer than. So DNA, and those are research papers, peer-reviewed and everything, they're talking about how unstable DNA is. All of a sudden, after that, in the 80s and then 90s, when PCR came out, where each cycle of PCR is heating up at 95 degrees of DNA, so it the double helix opens. all of a sudden DNA becomes a very stable thing, a very stable, chemical business. So here, I'm just going to read this quick. The main conclusion of this retrospective survey is that the body of examination examined theoretical and experimental evidence consistently and redundantly confirms scarce resilience and significant loss of structural integrity when DNA is heated at temperatures above 90 degrees Celsius, even for 1 minute. Such conclusion contradicts the claimed paradigm of PCR fidelity and raises the concern that at least for long sequences, if PCR can amplify some information, such amplified information may be unreliable for diagnostic or forensic applications, since it originates from sequences of nucleotides subjected to random fragmentation and reaggregation, because they actually are talking how after it fragments, it can actually also get together again. But the thing is this, Fabio just emailed me like a month ago. So this came out last year. He just emailed me a month ago, the same paper where it's published, it quarterly reviews of biophysics. So actually this is where it belongs, because it's physics of DNA that's being questioned, right? Like with all the things. They want to remove his paper. They actually wrote him because somebody, I don't have that paper pulled up, but somebody wrote a paper, and I understand why that's going on. Somebody wrote a paper where they completely ignore, I would put it like this, completely ignore all the findings that Fabio and Roberto are talking about from the pre-1950s or, the early 1950s, like that era before the whole thing, because I think something happened in the 50s. And we know that. I mean, viruses were confirmed in the 50s. So something was going on back in the 50s. So of course, this guy that published that other paper that's saying, based on my findings, this paper needs to be retracted. Of course, he is living in that paradigm where DNA is a double helix that's been proven to exist. We've isolated. Fabio and Roberto, they wrote reply to this whole thing, but the problem is, I told Fabio, I'm like, if he lives in the upside down world, and if the editor of the journal lives in the same upside down world, you cannot do anything. You cannot show them how it's upside down, because you just cannot. They need to see it. I saw it for myself. They need to see it for themselves, and that's it. So I'm still waiting on hearing what's going on, but just wanted to point this out because you were talking about breaking. So you were talking about breaking because of the X-ray, but I'm actually talking about breaking because of the high temperature. But still, it's breaking of DNA, right? So now just to go quickly back. So these are thoughts and conclusions that she draws at the end of the paper. I mean, the paper is long as can be, pretty much. So if somebody is interested, you guys can go read, because she is talking about all these different checkpoints, what she's questioning and all of that. But here, just a few questions I'm going to read. What makes scientists believe that the inactive part of the cell, because it's kind of talked about that nucleus is actually inactive, because in the cytoplasm, where you look it under the microscope, not electron microscope, but the light microscope, right? So you can actually see moving in the cytoplasm, but nothing is moving in the nucleus. So she's kind of wondering why something that's inactive would contain this vital information of life. And that's important for tissue formation and everything, right? What makes scientists believe that components extracted from chemically treated tissue of one species represent the content of all cells, nuclei and nucleic acid of all species? Why vital research papers describing the methodology of extraction of DNA and its component are not freely shared, with some being totally missing? How many scientists actually read them? So what I can tell nobody is reading old papers. And again, what I can tell even when students are asked to read Watson and Crick paper, they see it as waste of time. They definitely don't want to go read it. And they read it a little bit superficial, and they definitely do not catch the phrases we suggest, we propose, and so on and so forth. They don't see any of that, because once they are in that upside down world, they do not see these things, because they cannot even fathom that something can just be a model. Because we've been working with this double helix DNA for decades now. So they cannot, in 2024, 25, they cannot even believe that something like that could be the case, right? That it's still just a model and nothing more. Do scientists ever question the procedures and methodology they perform? No, we don't. We definitely don't. So the difference that I saw between how I was doing things back in the day in the lab, we were making our own what's called buffers, right? Like buffer where you have all those chemical components in, and we would go take the powder of magnesium, salt, weigh it out, how much, this and that. Right now, you follow the protocol, you follow a kit that you buy from a company. You don't even know what's in those buffers. You don't know what happens when you mix. Let's say you have different of these solutions, again, called buffers. You mix, let's say you're extracting DNA from bacteria. You have bacterial paste. You mix solution number one, which contains, I know what it contains and I know what it does. Students now, they have no clue. They just know they need to mix bacterial paste with that solution. Mix it for a certain amount of time, like vortex it, like really strongly mix it so it all dissolves, and then you put solution two. They have no clue what, even theoretically, what's going on. I mean, again, what I know is going on is just, again, what I was told is going on. Let's make that clear. But at least I was told something. They just do it, and they don't think about, and they don't question anything. And that's why I can tell, and this difference I noticed 20 years ago when I came to the US. In the US, it was much stronger, the following of protocols, compared to how it was in Italy. And that's why when 2020 came, scientists were told to put the mask on because, that virus. They didn't question it. The protocol, Dr. Fauci, whoever said mask will help you, they didn't question it, because they, over the last couple of decades, they learned how not to question anything in quote unquote science. So.
Yeah. I graduated a little more recently, right? I was year 22, and I can attest to exactly everything that you're saying, even in the way that we're being tested, because obviously we're given lectures and then we have exams and we must regurgitate the information to pass. Laboratory components are quite low in weight and in time in most classes. Of course, some are maybe like 50%. Some of them, I had a couple of full classes that were laboratory as well, but introductory ones, which when you're talking about getting into med school, you're not taking the 3,000, 4,000 level, in-depth laboratory courses that should be, assessing the methodologies, discussing the history, discussing the methods, but actually don't. It's still just step by step. That's what's leading, to their understanding. So very limited understandings. You'd be given, even in intro biology, or even in my 3,000 level biochemistry courses, you're given one lecture on history, maybe even half a lecture. And it really just depended on the prof and how much they were interested. And then come test time, the only thing you were tested on, who isolated DNA, who took the picture of DNA, who invented Koch's postulates? Was it Robert Koch or Edward Jenner? It's like hilarious, right? But there was maybe like two or three questions like that just to throw it in there, for the extra couple points for the students who studied every single lecture. But there was no emphasis on the methods, reading the papers. It was never even suggested to go read papers. A big problem now in, all of my schooling, even in my master's, we're being told to reference new papers. If you reference something older than 10 years, it's considered, old science and not, important anymore, which is just, absurd, right? The scientific method is a scientific method, regardless of when it was used. But this is what's being perpetuated, right? This is the idea that if it's not a new paper, then it's no good. And these new papers, just take assumptions as facts and portray them as facts. And so when you only read new papers, you only get these new stories, and it just furthers the problem, right? So that's the big problem with history, right? There's no attention to the methods being used, even the modern methods that are used, like even when we were in the lab, working in the lab with gel electrophoresis, you'd touch on the methods. But again, it was like not the important parts of it. I took a couple courses where it was talking about, the actual function of X-ray crystallography, of NMR, of these methods, right? And you would talk about the physics of it and, how the equations being used, but you never addressed like the philosophy of why you're doing it, how this adheres to the scientific method. You were just told like you're given this piece of data be based on the electron scattering. Like they leave out all the important parts that assume things, right? And they just assume it and continue on. It's so interesting, and obviously you don't question it when you're in the moment. The other really cool thing that you brought up was trusting the companies to give you valid, valuable, components to your reactions, right? We're just trusting the company's test kits and solutions. And a big problem is antibodies. And I know this is a little bit of an aside, but I want to throw it in here because I'm going to bring it back with a little point here at the end. Um, we trust, antibody research. Antibodies are the cause of the reproducibility crisis in biomedical literature, especially when it comes to things like our biological systems and testing the phenomenon thereof, like our germs or viruses or our immune systems or whatever it is, right? It's all due to antibodies. We're told that antibodies are specific. It's just not true. Empirical literature shows that that's not true. I have YouTube videos that you can go listen to that more in depth. Point is that there are so many papers out there that say that we're just too trusting, that we're receiving monoclonal antibodies, which are specific antibodies, right? And that we don't use controls because supposedly these are specific. So why would I use control if the company's telling me it's specific? I don't need to control it because it's specific. I don't need to question the method because the company told me the method is valid and my results will be good. This is what's told and what's believed, right? And so it's interesting. I'm building out a classroom on parasites for my community, and I'm talking about how they diagnose parasites in people. And the big ones are microscopy, right? That's the gold standard, supposedly. When it comes to protozoa, they say that microscopy is unreliable. So they rely on PCR and serology. So I really tried hard to find a paper. I'm like, can we look at all three side by side, test the same samples, positives, negatives, what's the correlations look like? There's so many methodological questions that arise even in this discussion. But when you use PCR as the gold standard, a positive or negative is like a true positive or negative. That's the gold standard. Microscopy is about 50% correct. It's a coin flip whether or not the diagnostic criteria was met through microscopy. It's worse with serology. You know, PCR and serology, they don't align. Microscopy and serology, they don't align because then they use microscopy as the gold standard. When they thought through their subjective interpretation of the samples through microscopy that it was positive, PCR dropped to like 50%. And then it was just a 50-50. And these papers are rare. Like I've only found a couple of them because they just completely dismantle the idea that there's any sort of validity to these tests. And these same exact tests are used in genomics, in the study of the genes, when it comes to GMO, when it comes to gene editing, when it comes to the genetic code, when it comes to diagnosing people based on their genes. It all comes down to PCR, right? But at no point has PCR been proven to be valid or a gold standard, right? We just said it was during COVID, and all of a sudden now it's the gold standard. It just is mind blowing. Like what can we conclude from this? What can we even say from this? It's just all nonsense. Like I don't even know what to conclude from it all. It's just that we have absolutely no idea what these tests are doing, what they're testing for. We don't have consistency in our answers whatsoever. We tell really convincing stories. People in power and the populace tell convincing stories and we believe it. That's what's going on. Look at the objective data. It's completely different. It is completely different from what's being told. And that's just not being done. The other thing that I noticed, last point here, was that when I was reading the old, human control, um, infection model studies back in the late 1800s, early 1900s, I was reading papers and I was trying to find these old exposure studies where they take a pure parasite, give it to a healthy person, and see if it caused disease. I was reading those papers. It was so hard to find these papers because at least 25% of the time, these papers were miscited. They were cited as being in the wrong journal or the wrong pages. And so it took me hours and hours to find these papers. When I finally did, I went and read them and it was like, oh yeah, no symptoms developed. And it's like, okay, there's such a disconnect. These papers are misciting, and there's like the whole literature does it. It's across the whole field of parasitology, of germ theory, of viral theory. When you go back to the original papers, they're being miscited. So they're obviously not reading it if they're incorrectly citing it. They read another paper that cites it, and then they copy the citation and they rewrite it, they paraphrase it, and include it in their paper. And there you go. It's like they're not actually going to read the original paper themselves. It's the same with the methods, it's the same with the theories and hypotheses, whether they've been validated or falsified. It's just not done. There's a huge disconnect in the true use of the scientific method and even the use of empiricism and scientific discussion. And it's not discussed at all.
I want to confirm everything you just said, meaning when we are citing papers. So what happens now? Everything is you go PubMed and you click and you get a lot of the recent papers, even like 20, 25-year-old papers, you get them in a PDF version, right? But then good luck finding papers from the beginning of 1900s, right? In a PDF version. So you kind of need to go get yourself maybe to some medical college, medical university library, right? Where maybe some paper that's not Nature, that's not the Lancet is available, right? With all this stuff that you're looking for written about. And so, yeah, so what we do, we usually just find somebody else that cited it, and then you cite it because that person cited it. But what was even written in there, nobody really knows. But you just cite it because and it's so true. And it's so true. I mean, yeah, it's at the end, it's really comical because people are just, and we know what we're doing. Like scientists know what they're doing. Scientists know the peer review is, as I said, a little bit BS, but they don't really connect the dots to understand what it really means for everything we're doing. Because if we are just citing like this, if peer review is nonsense, so what does that mean for my research? That my research is questionable, right? Because everything is connected here, right? So you touched on antibodies. And I want to point out something else because antibodies are used to determine all these pathways. I'm not pulling out the image. You know how the pathway looks like. It's all of these proteins connected, one interacting with the other one. And all of this stuff was discovered using this, discovered. Again, air quotes are just continuous, so I'm not even doing it. Because it's just constantly air quotes discovered and isolated and whatever. I mean, it's all based on these antibodies, and it's ridiculous. And we talk about it. Just a few days ago, I talked to somebody that's using antibodies, like fluorescently labeled antibodies for their imaging and stuff. And he's like, oh yeah, right. This technology, it's called geomics, is using fluorescent antibodies, but it's not the same. Even if it's for the same protein, it's not exactly the same antibody as the other technology that's called Orion is using. Because I'm like, I don't see the same result in these two images. Oh yeah, because this one is used, and I'm like, so even he came to the conclusion, yeah, right, so it's a little questionable. I'm like, yeah, it's a lot questionable. And they are a core facility, meaning they are the ones who are doing experiments for everybody, because I'm not doing all of this, like this new technology, somebody learns and then they get all these slides and do it for all the labs. But everything based on antibodies is a bunch of stuff that should never be touched with a 10-foot pole. I mean, we should just stay away from it. But then everything is based on that, on pathways and all of these different molecules that pharma companies design to block a certain pathway so your cancer won't develop. Again, it's all based on stories. And the fancier story you tell, the higher the chances that you get a good position in academia. I mean, Harvard or some sort of little college in rural, whatever, Tennessee, right? Or Vanderbilt where is Vanderbilt? Vanderbilt is in Tennessee, University of Vanderbilt. So, I mean, it really depends how your data look like and if it's fancy. So we went a little bit out of the way from DNA, but it at the end, it all connects, so.
Yeah, absolutely. Yeah. Like you're mentioning, the way that we understand how DNA comes out of the nucleus and then moves to the ribosome and gets translated into a protein. It's like that's all confirmed through serology, right? Through the use of antibodies, and beyond, right? So it's always connected. To bring it back, talking maybe now about genetic code. Now we have all these methods, metagenomic methods, Illumina, nanopore, sequencing whole genomes, and we know, all of these genes. Pretty foundationally, we're talking about the Human Genome Project. Part 2, where we discuss the Human Genome Project, gene editing, GMO, DNA testing, heredity tests, CRISPR babies, and more, will occur in the next episode.
All right, and I want to thank you all for listening. Before we wrap things up, please remember this is for informational purposes only. This is not medical or therapeutic advice in any capacity, and it does not replace the advice from a qualified professional or practitioner. But please remember, we are responsible, sovereign beings capable of thinking, criticizing, analyzing, and understanding things for ourselves. We and the greater forces are self-healers, self-governable, self-teachers, and so much more. I really appreciate all of you for taking the time to listen to the podcast today. If you have any questions, criticisms, comments, concerns about the podcast, please reach out to me on Instagram. It's probably one of the best ways. Email's fine as well. And of course, if you feel like something resonated here, if you learned something, please like, share, subscribe, comment, and consider leaving a rating or review if you're listening to the podcast version. Please do not forget that the Beyond Training community is live. I'm telling you, this is not a space that you're going to want to miss. It's really something powerful, something amazing, something tailored to each and individual. I took the attunement piece that practitioners should use with their clients and I've integrated into the community. It's not a one-size-fits-all. There will be free resources over there, so make sure you go join as a visitor, gain access. And if you want to go deeper, of course, that is what I love to do. First deep dive is on parasites. It's truly a whole classroom in and of itself, integrated into the community. Future deep dives will include cell biology, genetics, mold, really whatever you guys want to learn about. That's the bonus of being a founding member. You'll be able to shape the direction of the community. We really talk about it all: movement, lifestyle. We talk about reflections, and there are reflective prompts in there to ensure that you are on your own meaningful path. And of course, there'll be live Q&A and discussion calls. Go and join, become a founding member. There will only be 100 spots. Go and join the community. It is now launched finally. You'll be able to lock in that low price, complimentary onboarding call, shape the direction of the community. There's no reason not to sign up right away. If you're ready to unlearn, reconnect, and rebuild from the ground up, I invite you to step in to the community. Just remember, guys, There are two types of people in the world, those who believe they can, those who believe they can't, and they are both correct. Thanks for listening. Take care.